Hello everybody. I'm in the classroom again, lecturing to an empty room. I miss you guys, but it's kind of nice to be here. It's still a mess. It's a disaster here. One of these days I'm gonna have to come in and clean it up. All right, so a couple things I just want to mention before I get started on the topic for today. First of all, your unit packs, please make sure that you're keeping those up to date. I know that. Okay, so when I polled you guys to ask you about what things from the first week you liked and you didn't like, you like this, from what my takeaway from this was that you kind of like the lecture to an empty classroom type bit. It's so not high tech, but it seems to work okay. For now, as long as they keep letting me in the building, I don't know how long that's gonna last. The biggest negative that people said was they didn't like the group work. I get that. It's still only Thursday, so they're not due until Sundays. So I've only seen a couple of them. They've been really good, but okay, if that's difficult for you guys, I will in the future avoid doing assignments similar to that. But there were a few people that said they were not enjoying using the textbook or doing packet work, and I'm so sorry, but that's the way it's always been in here. So yeah, you're still going to have reading assignments, you're still going to have study guides. There's a POGO that goes with this that you're going to be working on. Keep up with that packet work. Keep in mind that we're going to be having a quiz on chapters 19 and 21, not next time, but coming up not too long from now, and it's entirely possible that it is possible. The really only main way I can do that would be to make that open notes. I can time it, I can give you guys only so much time to complete it, but having your unit pack done might be something where I'm going to be just like, okay, it's open notes, do it. That's likely quiz work. Okay, so with that said, you have already at this point finished cases 1 and 2 on the Teddy Graham Lab, and we haven't done cases 3 and 4 yet. We're not going to get those assigned today. That'll be next time those will be assigned. So you can hold off on that, and that lab won't be due for a little bit yet. We're going to go back today and go through that part 1 that we skipped over once we learn about how to work the Hardy-Weinberg equation today. All right, so this is in chapter 21 of your textbook, so that's a good reference for this. We call this population genetics. We also call this microevolution, because here we're looking at changes in a population over time, which is evolution, but we're only talking about changes in the frequency of alleles. So if you have a population and its collection of genes, we call the gene pool, and for a particular trait, you have so many dominant alleles and so many recessive alleles. If that percentage of dominant versus recessive changes, that's evolution. On a small scale, because we're not talking about the evolution of any new species or any new groups of species, any phyla or anything like that. We're just talking about changes in a population over time, and that is evolution. That's what we're dealing with here, is evolution on the very small scale. Okay, so gotta back up a little bit to Darwin here again and think about what Darwin came up with. Remember, was the mechanism for how evolution could work, not by natural selection. But in his idea, and the reason he struggled with it for like 10 years before he published it, was because he was trying to figure out genetics. It was a very big difficulty for him because, remember, Gregor Mendel's work wasn't accepted for 50 years after this whole work was being done. Remember, Mendel and Darwin were living at the same time. So at the time, it was a difficulty for Darwin because genetics and heredity definitely plays a role in natural selection, changing populations as time goes on. And so he was trying to figure it out for himself. Even after Mendel's work was done, we didn't see, you didn't know DNA, you didn't know genes and things like that. So there was only like discrete characteristics. You could see the proportion of the population that had red flowers and blue flowers or whatever, but you couldn't see genes until later on. By the 1940s, though, there we now at that point knew about the genetic basis of evolution and were able to, I guess, put together the things that Darwin was talking about and apply this modern genetics to it, and it just gave it more strength. All right, so keep in mind this was on one of our big question ideas. Individuals cannot evolve. Individuals cannot change in their lifetimes. If you start saying things like that, then we're going to start calling you Lamarckian. That's not the way it works. Populations are the smallest groups that can change. It's the unit of evolution. Natural selection is the main mechanism for change in a population over time. However, we're going to find here in chapter 21 that it's not the only way that populations can change over time. It's not, there are other ways to get evolution going. Okay. So there's a couple of different ideas about the rate of natural selection, the rate of change in a population. Gradualism is the idea that Darwin was thinking about, that you'd have slow, continual, steady change in a population over vast expanses of time. It was like what he was learning about with Lyell and the geography stuff that he was reading about. And so that's what he was thinking. However, around in the 1940s, 1950s, there was another guy named Stephen Jay Gould, and he came up with the idea of punctuated equilibrium, that natural selection can push populations very quickly following changes that happen in the environment. Drastic changes in the environment will push drastic changes in the population. So time-wise, it can go through kind of some acceleration points and then deceleration points. Neither one of these are necessarily true for all species all of the time. They're just kind of different ideas about the tempo for how changes happen in populations. Okay, so we have to define a population. Population would be a members of the same species that live in the same area at a particular point in time. And the gene pool, as I mentioned before, is the collection of all of the genes for that particular population. Microevolution, I've already mentioned this as well, would be the change in the frequency of the alleles in the population. So when you guys were doing your calculating of P and Q, remember P is the frequency of the dominant allele, Q is the frequency of the recessive allele. If that changed at all, in either case 1 or case 2, that's evolution that was happening to your population of tetragrams. Okay, so there's an idea that then also has equations that back it up that's called the Hardy-Weinberg theorem. The Hardy-Weinberg theorem states that a population will not evolve provided 5 conditions are met. Okay, and so these 5 conditions for a non-evolving population, getting your P and Q to stay the same as time goes on, no matter what. First of all, you need to have a very large population size. You might have noticed when I did my teddygram demo that it didn't stay the same as time went on, and that's because I was only dealing with a population of 20 teddygrams. That's not a very large population size. However, when we put together all of our group data, we haven't done it yet, so I haven't seen it yet, but when we put together all of our group data, some of that should even out, and we should end up getting a population that does not change as time goes on via just chance. And that's what was going on with mine. So a very large population size will eliminate just chance, which ones happen to survive, which ones happen to have babies, what the genotypes of the babies happen to be. That's all randomness, and that will settle out and even out with a larger sample size. You can't have any individuals moving into or out of the population, because if you did that, think about your teddygram population, and all of a sudden you have, okay, instead of having 20 bears, you have 25 bears. You'd be like, oh, Mrs. Gilmore, I have to recalculate P and Q now. Yep, you would, because you would have probably a change in those allele frequencies, even if it is just small. So no migration. You can't have any mutations, okay? So we can't have any new alleles. So you can't have happy bears, sad bears, and some other kind of bears. That would change P and Q then as well if you had another trait as well. So you can't have any mutations, which honestly, you're getting the idea here that no mutations. How is that possible, Mrs. Gilmore? Well, it isn't. So this is all very theoretical idea. Keep that in mind as we're going through this. But if you do have mutation, a mutation is going to change P and Q even slightly. You have to have random mating of individuals. Random mating or non-random mating and natural selection sometimes go hand in hand, where females will pick males that have particular characteristics. Sometimes those are because of particular adaptations that they have in the environment. But in order to have a non-evolving population, it has to be completely random which individuals are mating together. And then you can't have any natural selection. Okay, so no natural selection. All traits in the population are completely neutral. Okay, once again, is that something that you're ever going to run across in a population? No, not really. So our teddygram experiment, and these I should update those values. That's something from a previous, I've modified the teddygram experiment. It used to be 0.5 and 0.5. But the idea that you would have in a non-evolving population is whatever we end up with for generation 1, which I don't know if it's going to be 0.5 and 0.5. As an average, what did we end up with in after generation 1? And we just drew them on the box. If we have a non-evolving population, whatever that p-value is should stay the same, even if it's, say, 0.65 or 0.7. If you have a population that's at p is 0.7 and q is at 0.3, and that stays the same over many generations, that is a non-evolving population. Okay, so keep in mind that non-evolving population does not mean that the alleles are equal in their proportions in the gene pool. They might be, but they don't need to be. It just means that they're unchanging. So what happened in the Teddy Graham situation when I was doing this was that you didn't have a big enough sample size. And so the group data that we collected hopefully is going to minimize the changes that happen due to genetic drift, it's called, when we don't have a very large population. Due to just chance, those changes happen. Okay, so these are theoretical. But what we use them for is you use those 5 criteria for a non-evolving population to try to determine what is causing this population to change as time goes on. Is it a natural selection thing that's going on? Do we have genetic drift happening, which can happen if you have a population of endangered species, or if you have a population that's new, moving to a new environment, like some individuals that end up on an island and they weren't there before, things like that. What is the reason for this evolution? And then you can also measure it or use it to measure the degree of change. How fast is this change happening? We can quantify this using the equation that's coming up here. So the Hardy-Weinberg equation is used to determine the degree or how fast the microevolution that's happening for this particular allele. Okay, let me see if we're gonna, okay, now it is time for us to learn about this equation. All right, so far we haven't. So far we have learned about what P is and what Q is, and you've calculated those. You know those are just frequencies, which basically are percentages that you don't multiply times 100. P is the frequency of the dominant allele, Q is the frequency of the recessive allele. When you add them together, you end up with 1. That's all of the alleles. Okay, it gets much more complicated if you have multiple alleles, but we're not going to go there. But what you can do with this equation is you can calculate P and Q if you do not know whether individuals that have the dominant trait are homozygous dominant or heterozygous. So this equation is going to help us to make predictions based upon mathematical probabilities. Okay, so we'll be able to answer questions such as this. I'm going to come back to this. Let's go to the lab. No, that's not what we're doing today. Okay, so next we're going to go to the lab. All right. Bummer that we're not here to do this. All right, so here's where I would give you guys a piece of PTC paper to see if you can taste it. I think you've probably done that at some point already. You know that those little slips of paper that have this chemical embedded in them that some people can taste and some people cannot taste. And if you can taste it, it tastes bitter. I'm a taster. I think it tastes like if you get hairspray in your mouth, it tastes bad. So imagine that you're doing that right now. We don't have, I'm going to make up data for this. Bummer. Best we can do though. All right, so tasting is a dominant condition for PTC paper. Homozygous dominant individuals would be tasters, big A, big A. Heterozygous individuals are also tasters, and homozygous recessive individuals cannot taste this chemical. They don't have the receptors located in their tongue, on their taste buds, in order to do that. Remember that genetics is always going to lead to a protein. So what protein's involved? It's this receptor protein on the surface of the taste buds. It's absent in individuals that are homozygous recessive. All right, so what we're going to do is we're going to fill in this data here. Okay, so everybody can get your lab out if you don't have that out. Oh, no Cubs. It's opening day today. I'm wearing my Brewer stuff because it's opening day. I'm sad. Where? Sad about lots of stuff. Okay, so I'm going to need my calculator too. I wonder where that is. I can... Nah, hang on. Okay, I got a calculator. Okay, so let's say that our class population, let's say that we're dealing with a class of 24. And let's say that in our class of 24, let's say 6 people cannot taste PTC. So we're going to do the number here, and it's 6. And so then that would mean that out of our class of 24, we have 18 people that can taste the PTC paper. Okay, so next thing to do would be to calculate percentages. All right, so this is 75%, and then this would be 25% of the population is a non-taster. Okay, that could be, should have been able to do that one in my head. Okay, but the people that are tasters, we can't tell whether they're homozygous dominant or whether they're heterozygous. So we're going to have to use the Hardy-Weinberg equation in order to figure out P and Q. We don't need to do this in the Teddygram lab because we are able to keep track of which individuals of the SAD bears are homozygous recessive or homozygous dominant and which ones are heterozygous. We can tell that because of the missing foot on the homozygous dominant guys. But in this, the tasters, I don't know, you don't know whether you're homozygous dominant or heterozygous. So we're going to have to use an equation in order to figure this out. Okay, so there's a few things that we need to know about. I'm going to do a okay. All right, so this is what we already know. We already know that P is the frequency of the dominant allele. Okay, this is one of the things people confuse. What's the difference between P and then there's going to be something that's P squared. Q is the frequency of the recessive allele. Okay, we've already done that also. Okay, that we learned about in case 1 and 2 in the lab earlier this week. Since these are the frequency of the dominant allele and the recessive allele, and that's all of the alleles, P plus Q is equal to 1. Okay, that's what we got so far. Now we're going to add the Hardy-Weinberg equation to this. So the Hardy-Weinberg equation is P squared plus 2PQ plus Q squared equals 1. Okay, so now the different parts of this equation. P squared is going to be equal to the frequency of the homozygous dominant genotype. Okay, so it's the frequency of the homozygous dominant genotype. 2PQ is the frequency of the heterozygous genotype, and Q squared is the frequency of the homozygous recessive genotype. Okay, so be careful, genotypes here, these are genotypes, those are alleles. Those are the things that people tend to get messed up when they're working on Hardy-Weinberg. Okay, so we know that in our population, in our class, we know that in our population, in our class, we had 75% were tasters, and tasters are the dominant condition, and so we know that those are the homozygous dominant and the heterozygous individuals. 25% of our classroom population are non-tasters, and we know that they are homozygous recessive. Okay, so we want to know, let's say we want to know the frequency of the heterozygous genotype. Okay, we want to know the people in the classroom that are carriers for the recessive allele, but they also have a dominant allele, and therefore they are tasters. So we want to know 2PQ. All right, so we don't know what P is, we don't know what Q is, we have to figure those things out in order to plug into times P times Q. All right, the place where you're always going to want to start, I think always, I'm careful with always, but typically the place where we're going to want to start is with the homozygous recessive individuals, because we know that their genotype must be little a. So we know that the frequency of them is the Q squared. Okay, so if there was 25% of our classroom population that were non-tasters, that means that Q squared is equal to 0.25. Right? How are we going to get Q from that? You're going to use your square root button on your calculator, although I think that one's a perfect square, right? Yes. Okay. So Q is equal to 0.5. Okay, so that means that half of the genes in our gene pool are recessive alleles, and the other half then must be dominant alleles because P plus Q equals 1. So then that means that P is also equal to 0.5. All right, so if we want to know the frequency of now any of these genotypes, these two are the ones that we don't know about. We can calculate them now because we've got P calculated and we have Q calculated. So 2PQ, that was the frequency of the heterozygotes. We would take 2 times P is 0.5 times Q is 0.5. And so the frequency of the heterozygous individuals is 0.5. 2 times 0.5 times 0.5 equals 0.5. Okay, so we expect then that half of the class is heterozygous for this particular trait. 50% of the class, 2PQ is 0.5. So that's the frequency, so therefore 50%. All right, I want us to try one more of these. So one more of these together. So now let's go back, and this is the information that you can use to plug into the, let's go back, make sure we cover the lab before I move to the last problem. Why? Why are we not wanting to go? Too many of these are open. Hang on, I'm gonna pause us. This is craziness. Oh, there it is. Okay, I'm not pausing us. All right, so we've calculated P and Q now. Here's the North American population here. Notice that we aren't right on the North American population. We had 0.25 and 0.75, but first of all, I made up numbers, and then we aren't a truly representative sample. It would be a small sample size. There's a lot of reasons why we tend to fall off of the North American population, and if we were together, we'd just kind of do a little comparison and think about that with real data, but not made up stuff. Okay, let's try, no, not that. Go back here. So this is a time where this might actually be a useful thing, being able to use the Hardy-Weinberg equation. So this is a situation where we know about a genetic disorder, and you can tell its frequency in the population because you know that it's homozygous recessive, and so you can tell the frequency of the allele in the gene pool by using the Hardy-Weinberg equation. So the question, 1 out of every 3,000 Caucasian Americans is born with cystic fibrosis, which is a recessively inherited genetic disorder. The question for what percent of the population is carrier? So once again, we're calculating the 2PQ, we're calculating that. So I'm going to you to pause this and go through and do this calculation. Okay, so you've paused this, you've worked this. Let's see how you did. Okay, so what color is going to actually show up? I don't know. Blue might be a problem. All right, so we've got 1 in 3,000 Caucasian Americans is born with cystic fibrosis. So we know 1 out of 3,000 individuals has the homozygous recessive genotype. So Q squared is 1 over 3,000. So that's a tiny number. Oops. Okay. And we then need to calculate from that what Q is. So we're going to take the square root of 1 over 3,000. And we get 0.183. Okay, that's Q. So keep in mind that P is going to be all of the alleles that aren't recessive alleles. So if roughly 18% of the alleles in the gene pool are, oh, I missed a 0. Oops, that seemed too big. It did seem too big. Let's add a 0 here. 2% of the population. That makes or 2% of the gene pool. That makes so much more sense. Okay, about 2% of the gene pool is recessive alleles. So we're going to see about 98% of the gene pool is dominant alleles. So I'm going to take 1 minus 0.0183 to get what P is. P is going to be equal to 0.982. All right, there's some rounding there. Okay, so now we know the frequency of the recessive allele and the frequency of the dominant allele, and we want to know the frequency of the heterozygous genotype. So we want to know 2PQ. So we're going to take 2 times 0.982 times 0.0183, and that is going to be the answer to this question. So times 0.0183 times 2 equals 0.036. So, and then the question is percentage. What percent? So it would be 3.6% of the population is a carrier. Come on. I don't know. I can't get that guy erased there. All right, but it's 3.6% of the population is a carrier. All right, that's what I have here for now. We're going to be talking about some of those other ways that populations can change in the next installment, hopefully, of lecturing to the empty classroom. Yeah, keep an eye out on Google Classroom also for information related to AP testing. There's been some decisions about that made, but it's the dates and stuff like that haven't been released just yet. So I'm waiting to get the full picture to develop a plan to then relay that information to you guys. So just be on the lookout for that information. All right, thank you. Have a good day.